Hello all. I wanted to give you a rundown on the objectives for exam 3 and to try and provide some examples of when you will see these objectives or examples of what we've talked about in class that deal with these objectives. So in total we've covered 4 sections: molecular biology of microbes, viruses, antibiotics, and microbial evolution. And there's a lot of overlap between these various sections. So we began by describing the process of the central dogma, particularly focusing not so much on the details of replication, transcription, and translation, but rather what happens in these steps. We focused on a few of the major role players, such as the ribosome, the RNA polymerase, the DNA polymerase, and gyrase. You should be able to describe what's happening in each of these steps, and also types of proteins that you would expect to find in each of these. Second, we talked about how the structure of prokaryotes has contributed to differences in the central dogma. For example, the lack of a nucleus allows for transcription and translation to occur at the same time. Likewise, because of this, prokaryotes don't have intronic sequences, whereas eukaryotic mRNA may have those. We talked about the genetic organization, including how particular genes are grouped together, such as the lac operon. We talked about how genes can be regulated in their expression, and in particular, this example was the lac operon, and how we talked about both the ability of CAP to bind to cyclic AMP, and the ability of LAC-I to bind to lactose controls the expression of the LAC-Z, LAC-Y, and LAC-A genes. You should understand this basic mechanism of gene regulation. We've spent much more time talking about features like DNA mutations and how mutations can result in changes in phenotype. For example, more recently, we've talked about how certain types of missense mutations, ones that are between two amino acids that are similar versus ones that are more distinct from each other, like serine and alanine, can have quite a broad effect on, or can have quite a detrimental effect on host proteins. We've talked about gene mutations and horizontal gene transfer, and more recently how selective pressures like antibiotics or bacteriophage can either lead to a rise in antibiotic resistance or can contribute to, in the case of bacteriophage, a reversal of antibiotic resistance. And then finally, we talked about 3 different types of horizontal gene transfer and discussed the similarities and differences of these mechanisms as well as vertical gene transfer. You'll note that a number of these topics here are ones that we've come back to in discussing antibiotics and microbial evolution. So those that have featured most prominently, such as mutations, DNA sequencing to RNA to amino acid sequencing, horizontal gene transfer, and selective pressures all might play a prominent role in this exam. Secondly, we talked about a lot of basics of viruses, using bacteriophage as our primary model and discussing a bit more in detail about human viruses such as HPV, HIV, and influenza. You should be able to describe the function of the virion structure or the viral particle. What is its purpose? You should be able to describe the life cycle of bacteriophages, both the lytic life cycle and the lysogenic cycle. And you should be able to distinguish these two apart from each other. So what are the key characteristics that differentiate lytic and lysogenic life cycles? You should be able to describe how viral particles are different from cells, for example, their lack of a nucleus, their lack of ATP, and how their replication can differ. We talked about how some viruses, like influenza, have RNA genomes, and how this requires those viruses to bring along into the host cell with them their own polymerases, RNA-dependent RNA polymerases. I'm not going to focus so much on the effect of HPV, influenza, and HIV on their respective host cells, so you can just take 9.5 and cross it out for yourself. We're not going to focus on 9.6 either on this upcoming exam. For antibiotics, we've talked about the spectrum, the targets, therapeutic indices, and effectiveness of antibiotics. I would like you to know the antibiotics that we talked about in the chart that you put together for the first lesson. You should know the classes. You should know what they affect. And for some, like the beta-lactams and the rifampins and the quinolones, you should know what the target is. In other words, the rifampins block transcription. You should know that RNA polymerase is a protein required for transcription and that the rifampins inhibit its function. I'm not asking you to know the spectrum of an antibiotic, but given two antibiotics, a broad versus narrow spectrum antibiotic, that you be able to make a decision on which would you use to treat an individual. We also talked about characteristics of good or ideal antibiotics, and those included features such as spectrum, selective toxicity, high therapeutic indices. In other words, therapeutic indices are ones where the antibiotic has a high toxic dose and a low therapeutic dose. We also look at effectiveness here. You should be able to choose an antibiotic from a list of them or from a couple of them given features such as these and justify your answer given these features. Next, I'd like you to be able to analyze experimental results for antibiotic effectiveness. In other words, analyze broth dilution assays to identify mix or MBCs, perform disc diffusion assays to determine the zones of inhibition, and take the data from this to determine between two or more antibiotics which is most effective or which is least effective in inhibiting microbial growth. As mentioned already, you should be able, I haven't mentioned this already. You should be able to describe the different modes of antibiotic resistance, and we've talked about 4 of them. Efflux pumps or membrane rearrangement, that's one. And hydrolysis of the antibiotic, prime example here are the beta-lactamases, modification of the antibiotic, or mutation of the host, which connects us right back to the features of molecular biology of microbes. Finally, and we'll talk about this on Wednesday, you should be able to discuss some of the social practices that may lead to antibiotic resistance. And actually, this became Objective 11.3, and so we'll talk about that in some more detail. Finally, with microbial evolution, this kind of brings together the ideas of antibiotics, bacteriophage, and molecular biology. And we've talked about how different selective pressures can select, for example, antibiotic-resistant strains of bacteria. So you should be able to explain how environmental pressures can select for mutants, and how mutations can afford microbial populations a selective advantage in the presence of that environment. You should be able to discuss how bacteriophages like the OMK01 phage that infects Pseudomonas aeruginosa, how these phages create a selective influence to shift microbial populations. For example, make them more sensitive to an antibiotic. And then, as mentioned, we'll discuss this last point in class the lecture before the exam. So if you have any questions on these, please let me know. Do know that there will be a review session with Julia from 4 to 5 on Wednesday, and I'll be emailing you with some details on when I'll be available this week, just to give you a reminder of when you can stop by my office to ask questions. Hope to see you all in lab tomorrow, and I'll talk to you soon. Take care.